Imagine you're watching millions of engineered nanoparticles drift in a tiny microfluidic chamber, and you wish them not just to sit there passively, but to communicate, compute, and self-organize in time, just like enzymes in a living cell. That's the vision of synthetic enzymatic reaction diffusion networks among nanoobjects, the next frontier in nanotechnology, where catalytic surfaces talk to each other through chemical signals. Why can't we just stick microchips on nanoparticles? Traditional electronics and mechanics scale poorly below a few microns. You simply can't carve a transistor onto a 10 nanometer particle. Instead, nature shows us how cells do it. Enzymes anchored to membranes perform reactions, generating messengers that diffuse to other enzymes, creating feedback loops that control everything from heartbeat to metabolism. Question: can we copy that? Answer: yes. By immobilizing designer enzymes on nanoparticle surfaces and using custom small molecule substrates as wires, we can build entirely chemical circuits at the nanoscale. How do we localize reactions near surfaces? Imagine coating each nanoparticle with a different enzyme, say one that converts molecule A to B and another that turns B into C, but floating them freely won't guarantee interactions. Instead, we tether enzymes directly onto gold or silica beads, spacing them nanometers apart, so when B is produced, it's emitted right next to its target. Example: researchers have used DNA scaffolds to position hydrogenase and oxidase enzymes within a few nanometers, boosting the local concentration of intermediates and achieving cascade reactions that mimic mitochondrial processes. Which chemistry can we use to build these networks? We need reactions that are fast, switchable, and bioorthogonal, meaning they don't get hijacked by stray molecules. Ideal choices include kinase/phosphatase pairs that add and remove phosphate groups, proteases that clip peptides only when triggered, and designer dehydrogenases that produce fluorescent signals. By wiring these reactions into loops, positive feedbacks for switches, negative feedback for oscillators, we can recreate classic reaction diffusion patterns like waves, pulses, or stable gradients on a chip. What kinds of functions can emerge? Picture a swarm of nanoreactors that sense pH and release a therapeutic cargo only when a local concentration threshold is crossed. A chemical AND gate. Or imagine a chemical oscillator on nanoparticles that pulses drug release rhythmically, synchronizing across the swarm like fireflies flashing in unison. In the lab, scientists have already demonstrated tiny catalytic particles that self-assemble into rings or spirals purely through reaction diffusion signaling, hinting at programmable morphogenesis at the nanoscale. Why is this central to making synthetic life? Because function in biology isn't just about structure. It's about dynamic regulation and information processing. By mastering how networks of immobilized enzymes exchange chemical messages, we learn the rules of emergent behavior. How a simple feedback loop becomes a robust clock, how gradients guide self-assembly, and how switchable catalysts can enact decision-making. These lessons will drive the creation of truly lifelike nano-machines. Systems that don't just exist, but that live, adapt, and perhaps one day reproduce. Enzymatic reaction diffusion networks leverage cascades of enzyme-catalyzed steps coupled with molecular diffusion to process information and generate spatiotemporal patterns. In living cells, such networks underlie processes like morphogenesis, circadian rhythms, and metabolic oscillations. Recreating these networks synthetically on nano-object surfaces promises dynamic behaviors, pulses, waves, logic gating. Without microelectronics, achieving this requires precise spatial organization of catalysts and tailored substrate pathways to mimic biochemical signaling. A surface patterned with glucose oxidase and horseradish peroxidase can produce traveling fluorescence waves when supplied with glucose and a colorimetric substrate. It's like arranging dominoes in a circle so that knocking over one triggers a rotating wave of falling pieces, But here the dominoes are enzymes, and the fall is a cascade of chemical conversion. Confined reaction zones at nano-object surfaces enhance local substrate concentrations and restrict diffusion, sharpening spatial control. Surface immobilization also protects enzymes from bulk deactivation and permits reuse. Techniques such as covalent binding, affinity tags, or encapsulation in porous shells enable stable attachment of catalysts. However, ensuring uniform coverage and preserving enzymatic activity upon immobilization remains a delicate balance. Gold nanoparticles functionalized with thiolated biotin link streptavidin tagged enzymes into dense, active monolayers. It's like planting seeds in a garden bed bordered by hedges. The walls keep the seeds from drifting away while allowing them to sprout and nourish the soil locally. Chemical networks need components that are orthogonal, reacting only with intended partners, and exhibit switchable kinetics or allosteric regulation. Cofactors like NADH/NAD+, pH-sensitive groups, or photoresponsive moieties can modulate enzyme activities on demand. Synthetic analogs of natural second messengers, e.g., caged ATP, offer external control knobs to trigger network states. The chosen chemistry must balance robustness, reversibility, and minimal crosstalk in complex mixtures. Light-activated caged substrates remain inert until a UV pulse frees them, initiating a controlled enzymatic wave. It's like using color-coded keys to unlock specific doors in a building. Only the right key opens its matching lock. Enzyme and some keys can be stored securely until needed. Networks are built by arranging modules, oscillators, amplifiers, logic gates that communicate via diffusive intermediates. Each module consists of enzymes whose kinetics and diffusion links are tuned to produce desired dynamic behaviors. Connecting modules requires matching their timescales and concentration thresholds to avoid signal mismatch or noise amplification. Computational models guide the design by predicting emergent spatio-temporal patterns before experimental implementation. A ring of 3 enzymatic modules arranged sequentially can generate sustained chemical oscillations around a nanoarray. It's like wiring programmable LED strips. Each segment responds to the previous one's signal, creating traveling light patterns when the timing and brightness thresholds are correctly set. Dynamic behaviors require catalysts whose activities can be toggled on and off or modulated in rate. Techniques include allosteric effectors, photoswippable cofactors, or competitive inhibitors that bind reversibly. By programming timed additions or light pulses, one can choreograph reaction phases, initiation, propagation, termination, across the network. This temporal layering mirrors cellular clocks and pulse generators, enabling synchronized behaviors among dispersed nano-objects. An enzyme fused to a light oxygen voltage domain becomes active only upon blue light illumination, allowing temporal gating of its catalytic step. It's like fitting a faucet with a motion sensor. What air? Water flows only when the sensor, light, or effector is triggered, then stops when the signal ceases. Nano-objects exchange chemical signals via diffusing intermediates or through direct physical contacts in crowded environments. The communication range depends on diffusion coefficients, network geometry, and local consumption rates of messenger molecules. Tuning these parameters allows creation of spatial gradients, quorum sensing behaviors, or synchronized oscillations. Effective communication thus transforms isolated nanocatalyst islands into a cohesive synthetic tissue. An array of enzyme-coated nanoparticles exhibit synchronized fluorescence pulses when their spacing allows diffusive coupling of a shared substrate. It's like fireflies flashing in unison across a field. Their light signals travel through the air, coordinating the group's rhythm. By combining modules, one can engineer functions such as edge detection, pattern formation, or environmental sensing. Networks can compute Boolean logic, generate timed drug release, or self-repair by catalyzing protective chemistries at damage sites. The interplay of reaction kinetics and diffusion gives rise to behaviors that no single enzyme could achieve alone. These emergent properties point towards synthetic analogs of cellular decision-making. A two-enzyme network transforms a linear substrate gradient into a localized pulse, effectively detecting the gradient's midpoint. It's like a crowd forming a wave in a stadium. No individual fan decides the global pattern, yet collective motion emerges from simple local rules. Maintaining network fidelity under fluctuating environmental conditions, pH shifts, substrate depletion, temperature changes requires robust enzyme variants and buffering strategies. Scaling from single nano-objects to macroscopic materials demands uniform enzyme loading and controlled diffusion pathways. Preventing fouling by reaction by products and preserving enzyme activity over long durations remain key hurdles. Addressing these challenges is essential for practical applications in smart materials and nanorobotics. Embedding enzymes within protective polymer brushes enhances their operational lifetime by excluding proteases and radicals. It's like coating steel beams with anti-corrosion paint. The protective layer preserves structure even under harsh weather. Deciphering how dynamic function emerges from enzymatic networks will illuminate the principles underlying cellular information processing. Integrating genetic circuits, ribozyme networks, and synthetic organelles could lead to fully autonomous nanosystems capable of adaptation and evolution. Advances in directed evolution, microfluidics, and single molecule detection will accelerate network optimization. Ultimately, mastering synthetic reaction diffusion control at the nanoscale paves the way to constructing lifelike systems from the bottom up. Researchers aim to evolve enzyme assemblies that self-organize into metabolic cycles, sustaining their own resource regeneration. It's like teaching Lego bricks not only to snap together, but to reconfigure themselves into new structures in response to the environment.